Have you ever wondered how space travel affects our bodies? While many believe that time slows down in space, leading to a potential delay in aging, recent research reveals a startling twist. Space may actually speed up the aging process in human cells. This intriguing finding raises significant questions about the long-term effects of extended space missions on astronauts' health. Catriona Jamieson, a stem cell biologist at the University of California, San Diego, aptly describes space as the ultimate stress test for the human body. Her team has made groundbreaking discoveries, indicating that the unique conditions of spaceflight can accelerate the aging of human blood-forming stem cells. In their study, published in the journal Cell Stem Cell, they observed that stem cells exposed to about a month in space exhibited diminished self-renewal capabilities and clear signs of molecular aging. This research underscores the potential risks associated with prolonged periods in space, which could have serious implications for future space exploration. To delve deeper into the effects of space on human biology, NASA conducted a fascinating experiment in 2015 involving twin astronauts Scott and Mark Kelly. Scott spent nearly a year aboard the International Space Station, ISS, while Mark remained on Earth. The results were striking. Researchers noted significant differences in their molecular profiles, including telomere shortening and DNA damage in Scott's cells, some of which persisted even 6 months after his return to Earth. This experiment highlighted the profound impact that space travel can have on human health. Jamieson aimed to explore these effects at a more granular level by focusing on hematopoietic stem and progenitor cells, HSPCs. For her latest study, her team collected HSPCs from the bone marrow of individuals undergoing hip replacement surgery. They then cultured these cells for up to 45 days, either in the microgravity environment of the ISS or in a controlled laboratory setting on Earth. To assess the aging process of these cells, the researchers employed various techniques. One innovative method involved using a fluorescent gene reporter, which allowed them to observe the cell's activity in real time under a microscope. They discovered that HSPCs in space exhibited heightened cell cycle activity, characterized by increased division and reduced dormancy. These indicators suggest that the cells were aging more rapidly compared to their Earth-bound counterparts. Upon the return of the cells from space, the team conducted further analyses, including whole genome and RNA sequencing, to investigate changes in telomere length, genomic stability, and gene expression patterns linked to aging. Consistent with the findings from the twin study, they noted a reduction in telomere length and a significant decrease in gene expression related to telomere maintenance. Moreover, Jamieson's research revealed a concerning trend. Stem cells that traveled to space exhibited a higher frequency of single-base mutations, signaling potential genome instability. Utilizing a computational tool named AlphaMissense, the researchers predicted that these mutations could have harmful consequences. They found that the space-flown stem cells accumulated mutations associated with clonal hematopoiesis, a condition where mutated blood cells proliferate and may lead to acute myeloid leukemia later in life. Notably, such mutations were absent in the cells that remained on Earth. Looking ahead, Jamieson and her team, in collaboration with NASA, aspire to develop improved methods for monitoring molecular changes in astronauts in real time. Their goal is to identify strategies that could mitigate or even reverse the harmful effects of space travel. Jamieson is optimistic that advancements in space research will not only enhance our understanding of human health in space, but will also yield benefits for people on Earth, as she notes that these studies have accelerated technological progress, making ground-based research more relevant to our health needs. So, What do you think about these findings? Could the risks of accelerated aging in space deter future space exploration? Or do you believe the potential benefits of space research will outweigh these concerns? Share your thoughts in the comments.